Okay, should work.
I've spent all morning doing the notes and writing down all the things that were said more than once.
Okay, that's
that
good.
one's important.
That's good. That's good.
So I have 6 pages of
Awesome.
things.
I'm glad to hear that.
Do you have a plan? Or do you
Yeah, I was just gonna go through everything and then we could do practice problems at the end.
I love that.
Problems at the end. Okay. So, first things first, what are our basic, let's categorize our amino acids
Okay.
by single letters. Okay. Let's start there.
Okay.
So for these, let's do all of the associated for the basic, acidic, and aromatic. And these, let's just do, this there's only one. These, let's just do, well, actually there's 2, but these just do a
I
couple
don't think I know the
examples.
bottom one.
Okay,
What does that even mean?
so the bottom one is referring to the beta sheets.
Oh,
When
yeah.
you had these turns.
Oh, we talked about this last
Mhm.
time. Okay.
Yeah. Okay, let's start there. So what are our basic?
Basic is K and, or no, D and E, sorry. Right?
You're flipping them.
I'm flipping them. K, R, and H.
Yes, and what do they stand for?
Lysine, arginine, and I don't remember that one. H. Histidine.
Yes,
Duh.
okay. Yeah,
It's
okay,
the
let's
only
go.
H.
What's that?
I said it's the only H.
Yeah. There's only one of everything.
D and E.
Yes, which stand for?
E is the one that is weird. Glutamate,
Yes.
and that's the one with the 2 dashes, right? Like the O's have dashes. 'Cause
They
go
can.
to
It's
either
the
one.
resonance
Okay.
structure, yeah.
Okay.
Yeah. And D is?
Aspartate.
Yes, awesome. Okay, let's do the aromatics.
Is that just the rest of them?
And that's the ones that have rings.
Oh, okay.
Mm-hmm.
Proline.
Well, not that ring, like has to have a benzene ring.
Oh, so like phena, whatever.
Yes.
F.
Uh-huh.
Tryptophan
Yes.
and tyrosine.
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 Yes. Okay. Well, let's do 3 polar and 3 non-polar.
Okay. Hold on.
Let's do polar, like uncharged on these.
Okay.
So they can't have a charge, so it's not anything we've already said.
Serine is polar.
Yes.
And TH, what is that?
Threonine.
Yeah, threonine. Would cysteine be non-polar?
Cystine's polar
Okay,
because of the sulfur hydrogen.
okay.
Okay, that's good. Let's do non-polar.
Not aspartate.
Well, yeah, aspartate is definitely polar. Well, aspartate is definitely polar, yeah.
Yeah.
Yeah. And aspartate would be E. It'd be
Acidic.
yeah, acidic. I can't remember the word name of the other one.
Okay.
Anyways, it's arginine is what I was thinking of. Anyways, non-polar.
Would be isoleucine?
Yes, isoleucine is one.
Is leucine one?
Yes,
Okay.
leucine is one.
Is alanine one?
Yes.
Yes!
Okay. The beta turns. What amino acids do you most likely find in the turns of the beta sheets?
Oh, we talked about this, but I don't remember which one it was. It was a fancy one, wasn't it?
You already said it earlier. When we talked.
Proline?
Yes. Proline is one. There's only one other iod option.
Okay.
It's the simplest amino acid. It's achiral.
Glycine?
Yes, glycine.
I almost said glutamate.
Yep. Okay, there you go.
Taking a picture so I can write this all down.
I'll send the video in the chat as well
Oh, yeah.
to upload it, so you can rewatch.
And listen.
Okay. Yep. Absolutely. Okay, now let's talk about our interactions.
Okay.
Let's talk about the non-covalent interactions we need to know. Interactions. So what are the 4 non-covalent interactions that we will be tested?
Electrostatic,
Yes, electrostatic is one.
van der Waals,
Yes.
hydrogen, and hydrophobic.
Awesome. And let's do an example of each of these.
Okay. Wow. Going ham.
Wow. Okay, let's do an example. Electrostatic would be
Hold on. Force charge between atoms can attract and repulse.
just give it a simple example. You
I
know.
don't think I gave. Well, I might be. I didn't put one on here, but
Let's do what it definition, sorry, and example.
okay. For definition, I said force between charged atoms,
Mm-hmm.
and then attractive and repulsive.
Forces between charged atoms. And what is an example of an electrostatic interaction?
I don't know.
Just think about it. So what are the charged amino acids we have?
We just went over it.
R, K, H, D, and E.
Yeah.
So if an electrostatic interaction would be a interaction between one of the positive and one of the negatives.
So like an R and a D.
Yeah, so like an R and a D. Amazing.
Okay.
Okay, van der Waals.
Van der Waals, I said very weak non-covalent interaction between dipoles of nearby atoms. Transient dipoles? What does that word mean?
That's just a fancy word. Transient. Like it's there, but it's not there.
Okay.
Yeah. Weak, and the most important thing, it's only happens in very small distances of atoms apart. So I'd say less than 3 angstroms. If I had to put a number on it.
Okay.
So what could be an example of van der Waals forces?
For my examples, in this, you want like dipole? Is that what you're talking about? Or
Yeah,
you
just
want,
like Jenny example, yeah, dipole would work.
I said packing hydrophobic side chains.
Mm-hmm. Hydrophobic side chains are great.
Okay. Hydrophobic meaning water doesn't like water.
Yep. Okay, let's talk about H bonding.
Okay, I said weak attractions between a hydrogen atom and another electronegative atom.
Mm-hmm. More. Here you go.
Bonded to electronegative atoms like N or O.
Or S.
Or S.
Yes. And then the example of that would be DNA and then disulfide bridges.
Yes.
Okay, let's talk about hydrophobic effect.
Okay. Hydrophobic effect, water fearing, normally nonpolar, found in protein's core.
Mm-hmm. So, non-polar atoms face in, polar face out, forming blobs.
Blobs?
Yeah, I mean that's just what I call them.
Okay.
So like in a water cage example.
Oh, like the oil and
Yeah.
yeah.
And then this is polar, and it's water out here. H2O solution. Okay.
My example, I said oil and water.
Yeah, that's a
Okay.
great example. So yeah, anything that's non-polar. It could be
Okay.
fats, it could be oils, et cetera. Okay, great.
Okay, you can erase it.
Okay, sounds good.
Since there's the video.
Awesome. All right, let's talk about. Is it still recording?
Yep.
Okay, cool. Let's see, what's next? I'm trying to think. Okay, let's do. I'm just dropping everything. Finding charge.
Oh yeah. Like net charge?
Yes.
Okay. I actually just explained this to somebody.
Specific pHs. Awesome. So you're basically an expert.
Yeah.
Okay.
The only thing that's going to get me on this free response is going to be drawing the amino acids because I know I'm just going to mess something up. But as soon as it tells me what amino acid, I know I can find the net charge from it, but
Right.
it's going to be drawing it.
Right. Well, that's good, but what's the rule that we have?
If the pH is less than, then it's protonated. If it's greater than, it's deprotonated.
What if it's
Did I say it
equal?
backwards? Okay. It's 50-50.
Yes. No, you said it right.
Okay.
You said it right. Awesome. So 50-50. This is depro. Pro. Okay. Cool. So now let's find the net charge of this amino acid I'm going to draw.
Okay.
So Y.
Okay.
And we're just imagine there's an N terminus here
Okay.
and a C terminus here. At 2, 7, and 14.
Can I come up there?
Yeah,
Okay.
you can.
Can you- Ow!
Just explain the steps as you do it.
Okay. I'm gonna erase these two because we
Why is
don't
that?
even have to pay attention to them because they're not acidic or basic.
Yes. And the one she erased was lysine and tyrosine?
Yes. H stays, D, E, and K stay.
Mm-hmm.
Okay.
What's the next step?
Then I need to know their pKa's.
Yes. It's 3, so 3, 11, 4, and then 8, which these will be provided to you tomorrow, so you don't need to memorize those.
These?
The PKs
Oh, okay.
of everything.
Do I have to include these?
If it says you need to include them, you need to include them.
Oh, I've never done that before.
Well, you might have to. It depends on what he puts. We're just doing it there for
Okay.
the sake of math.
So like the N term
Mhm.
is, the pK is what? Or the pH, what are we finding it at?
Oh, 2, 7,
Oh.
14.
So the pH is less than, so it's deprotonate. So the N term, would I consider acidic or basic?
What in term is acidic? Sorry,
It is?
basic. Because it's positive and it's NH3 plus.
And this one
is
would be acidic.
Yes.
Okay. It's basic and it's deprotonating, so it's +1.
Is 8 bigger than 2? This is the PK, remember? So
Oh,
it's protonated.
protonated, so
Yes.
it's +1?
Mm-hmm.
Okay.
Just writing acidic for the reminder for the people watching.
The same thing, RHK, D.
Yeah, here you go.
Okay. D is acidic, so it's technically, I think of it like minus. And this is here, so it's +1.
Well, D's acidic, so if it's acidic, would it be giving a charge?
Oh no, it'd be minus, or just be 0.
Yes.
Okay.
Yes.
E is acidic.
Did you do N term?
Yeah.
Did you do H?
That's what I just did.
Oh, I thought you were doing D. I'm sorry. You did +1. You did it right. I'm
Okay.
sorry. I'm sorry. You did it. My bad.
D is, what was our pH? I'm sorry. Oh, this is our pH. This is our
2 is our pH.
I need to ignore this. 3.
Yeah.
Our pK is higher than our pH, so we protonate. D is acidic, so it would be 0.
Yes.
E,
The same thing as
same thing. And K is positive in this one, so we deprotonate, so minus 0.
K is the pK, 11 is the pKa, the pH is 2. Is 11 bigger than 2?
No. Gosh.
Yeah, 11's bigger than 2.
Oh, yes.
Yeah.
So protonate.
Yes.
But it's a basic, so it'd be +1.
Yes.
Okay.
So your net charge is
3.
+3. Charge it down. Make sure. And we can cross-check. This makes sense because this is an acidic solution. So the polypeptide balances by turning basic.
Okay.
Okay, let's do 7.
7 is less than 8. So these are our pH.
Yes.
Okay. My brain needed that.
You're good.
pH is greater than pKa. So it's deprotonate and it's an H.
The pH is greater? 7 is bigger than 8?
I missed this one, so I'm
Okay,
starting
you're
with
good.
6.
Okay, we'll start with the H. Okay. Yeah, 7 is bigger than 6. Yeah, so it's deprotonate, yes.
Oh, and I also missed this one on one. Let's just back up. I missed C term on
Oh,
the last one.
I missed it too. But it would still be +3.
It would, because it'd be 0.
Yeah, because this is deprotonated. So
Okay.
+3 is the right answer there. So it'd be +1,
0. No, +1, +1,
0, 0, +1, 0. And then you can just put on the side or something.
Equals 3 plus 3. Okay. 8.
Mm-hmm.
8 and 7
Mm-hmm.
are pKa's is larger than our pH. It's protonate. This is a basic.
Mm-hmm.
So it would be +1.
Yes.
7 is greater than 6, so it's deprotonate. Our H is a basic, so it'd be -1. No. 0.
Mm-hmm.
Because it's basic. 7 is greater than 3, and D is acidic, so it'd be -1.
Mm-hmm.
Same thing. And 7 is less than 11, so it's protonate.
Mm-hmm.
K is basic, so it's +1.
Mm-hmm.
And then acidic, 4 is less than, so it's protonate, so it'd be 0.
4 is less than?
4 is less than 7?
4 is the pKa.
Oh,
yeah.
the pH is greater, so it's deprotonate, so
Yeah.
-1?
Yes.
-1.
Mm-hmm.
Okay. 14 is greater than 8. Our pH is greater, so it's deprotonate a basic -1.
It's a basic. Why is it -1?
Because it's O, so it'd be 0.
I'll write this down here.
If you're deprotonating an acidic, it's -1, but if you're deprotonating, yeah.
Mm-hmm. There you go. Oh, wait. 0,
0.
sorry.
Okay. 14 is greater, and it's a basic, so it'd be 0. D is acidic, and it's greater, so it'd be 14 is greater. Deprotonate -1. 3, same thing. 11, K is basic, and it's still greater, so it's deprotonate, so it's 0. Acidic, deprotonate, -1. Equals -3.
Yes. Amazing. Okay, good work.
Sorry, I got confused here and there.
No, all good. All good. So, on the video, for everyone watching, writing these down right here, this table, and then this is going to be your key to these problems. If you know this, you can solve everything else just by looking at it. So memorize these two tables. Okay. With that being said, we're going to do the same manner, except it's going to be a drawing problem now. Here's my good eraser. Okay, so we're gonna draw a polypeptide. Let's do Y S I don't know, K. So when it says draw a polypeptide, it's already implied that you have to draw the N terminus and C terminus. If it just gives you a strand, it's not implied, so you can ask, do I have to include the N and C? But if it says draw, you have to draw N and C termini. So let's do YSK. Let's draw it at a pH of 2. Actually, I want to change it. Let's do it at like 4. And then we'll find the net charges of it at 4, 8, and 13. So you can just draw this one along and I'll draw it here.
Okay.
Okay. So the pattern to draw a polypeptide, as a reminder, it's going to be N, R standing for side chain, and carboxyl. And the very first N is going to be the NH3 plus, or H3N plus, since you're drawing from the left side. That's the N terminus. And the very last carboxyl will be COO minus. So I'll draw it, and everyone, you can draw along as well. Okay, have you drawn it?
Mm-hmm.
Okay.
Wait, why'd you put OH at the end though instead of COO?
That's the same thing.
Oh.
You could draw like this or CO minus, it doesn't matter. Okay, now we've just drawn it in regular. Now the next thing we need to make sure it's at a pH of 4. So that means we need to take off hydrogens. So Y doesn't matter, S it doesn't matter, but K does matter because it's a basic. It has to be one of these 1, 2, 3, 4, 5, 6, 7 things for taking off hydrogens. And that's how we do this without drawing it. So H3N plus, we know it has a pKa of 8. 8 is bigger than 4, it stays protonated. Let's look at our K. We know it's 11. 11 is bigger than 4, it stays protonated. Then we have our C terminus. And then the C terminus, I mean, it's about 3 point something, so it'll deprotonate. And it'll be a negative charge. So the net charge of this at pH 4 is +1. So now let's do those calculations for 8 and 13. And I can modulate the drawings as we go too. So 8, we're only looking at the N terminus, our K and our C terminus again. 8 and 8, so this will be 50-50. So thankfully you don't have to draw this, but I'll just draw like, I don't know how to draw half, sure. So it'll be 0.5 plus 11 is bigger than 8, still plus 1 here. And then 4 or 3.8 or whatever is smaller, so it'll be a minus. Our net charge is plus 0.5. And this is still how you would draw it, just this would be negotiable. Now, is that cool? Sorry to move on. Okay, now our last thing is 13. So 13, this is definitely going to be deprotonated, which means it loses an H. Then we have our NH3 plus. It also loses an H and the charge. And this will stay deprotonated, so -1. Any questions there?
Mm-mm.
Cool. Okay. And that is how you do those problems. Okay, now let's do separations. First talk about purification techniques. So let's say I have this jar. This jar has protein, DNA, and I don't know, other stuff. And I want to finally figure out what's in this protein. Let's just walk through what the steps are to get there, and then we can do a more relevant problem or more specific problem. So to get this and just get to the protein, what are the first two things I could do?
You can do a charge separation, right?
SDS page.
Yeah.
Is that what you're looking for?
Wait, no, not size. Like anion.
Not yet. We just have a jar of stuff.
Okay.
This isn't just pure protein. You can't do that until pure protein. That's important.
Mm-hmm.
So you have to do SDS page.
Okay.
And thinking.
Mass.
Mass spec is for proteins by size, right? Yeah. Okay. So that's SDS page. So now let's just draw it out. So now we have a jar. Now it's separated. This much is DNA, this is stuff, this is protein. So now we can take this part and we can separate it by weight via mass spec. Or gel filtration. Or the other way you learned it is by charge, which is cation, anion, exchange, chromatography. Okay. So just as a brief reminder, in gel filtration, what comes out first?
Whatever is not attracted to the...
It's not a charge.
Oh.
Gel filtration is by size.
Okay. So the smaller one? Or the large one?
Yes.
And then medium and then small?
Yes. Because our gel is shaped like this. It has the pores where these stick.
Mm-hmm.
Okay. Now let's do cation, then anion.
Cation binds positive charge protein, so negative comes out first.
Yes. Negative to positive. So what is the gel? Is charged?
It's charged cation positively.
The gel will be negatively charged
Yeah.
because it attracts to positive.
Mm-hmm.
Okay, and about anion is the exact opposite. So positive, then negative.
And the gel is positive.
Gel is plus, yes. Charged. Okay. Now, how do biochemists in gel filtration get the small proteins out of the pores? or get the positive and the negative out of the cation and anion? How do they remove it?
You have to add a solution, which is either add a solution or you said something else in class, but I can't remember. Salt bridge? I think no.
NaOH.
Yeah.
Which is a strong base. So add strong base is the answer there.
Okay.
Okay. Cool. Now with that knowledge, let's do a practice problem. So we have a tub of, I don't know, lysine at 800 Daltons. And we have 50, then let's just do like D at 400. So let's do the order of elution and gel filtration, then cation, then anion. We still recording?
Mm-hmm.
Okay, awesome. I want to make sure. So what would elute first in gel filtration?
Gel filtration is not charged, it's based on the size.
Mm-hmm.
So the 800 lysine,
Mm-hmm.
and then D,
Mm-hmm.
and then L.
Yes, because it's largest to smallest.
Mm-hmm.
Okay, how about cation?
It would be negative first, which would be D and just D, right?
Mm-hmm. Then?
And then L and then K,
Mm-hmm. Why L first?
because it's a neutral.
Yes. Okay, and how about anion? It's the exact opposite.
Yeah.
So it should be K, L, D. So this is positive to negative. Okay, cool. Now that we got that, let's do Henderson-Hasselbalch. Did he say he's gonna give that to you on the exam?
Honestly, probably.
Okay. What is the equation though, just so we know it?
Do not remember.
Okay. pH equals pKa plus log.
Deprotonated over protonated? That one?
Yes.
Yes, this is gonna be on our exam.
Okay.
It
Well,
was
just this is a math problem. So
Yeah.
let's just say we have a protein. There's 10,000 millimoles of it, dpro, and then there's 0.1 millimoles of it, pro, at a pKa of 6. So what is the pH?
You would do 1 times 1, 2, 3, 4.
Let's
and
just
then your 4.
just write it out. So yeah, so first thing is substitute. .1, 10,000. And we can ignore these because they're the same. If these units were not the same, you have to convert first to the same unit, preferably moles. So yeah. Okay, now we have to divide 10,000 by 0.01, which would be 100,000. We have to simplify the expression of the log expression, which we can do here, which we can say that x equals log of 100,000. This is a 10, we can do 10 to the x equals 100,000. So x equals 5.
So 6 plus 5.
Which means the pH of this would be 11. Any questions there?
Mm-mm.
Okay. I think I got all the hard stuff out of the way. What's next? I can't think of anything off the top of my head. What would he put? Is there anything on the review sheet that we haven't talked about?
Can you go over orthologs and paralogs? Like,
Orthologs and paralogs, yeah. Go ahead.
like when we're like talking about sequence alignments and stuff, is it just when it tells you like in the like problem, if it were to be like in 2 human things, you'd be like, oh, that's a ortholog because it's, or that's a paralog because it's the same species, but if it were to talk about different species, that's when you'd say it's a paralog? So like the only way to
Yeah, what are the definition of these? I honestly don't remember. It wasn't just
genes in different species that come from a common ancestry,
which is?
orthologs.
Okay. So the species is different, common ancestry, okay.
And then paralogs is same species that arose from gene duplication.
Good. Okay. So what was an example? So I mean, I guess
Hemoglobin gene in human and mice. Ortholog.
hemoglobin gene and human and mice?
Uh-huh.
What does he mean by human and mice?
Like they're different species, but they have common
Like
ancestry.
hemoglobin in mice?
Yes.
Oh, okay.
Like
Yeah.
hemoglobin in humans and hemoglobin in mice.
Oh,
would
yeah,
be an
yeah.
ortholog.
Okay. That'd be a good example, yeah. HG in human and mice. They're similar but different. So another thing that we could think of is, I don't know, our heart and a bird's heart. Those would be orthologs. Now paralogs.
Human hemoglobin, alpha chain, and beta chain.
Yeah. So he's using alpha chain and beta chain because they're the same gene, but they do different things. So for example, like best thing I can think of is like skin.
Yeah.
Because like people who live by the tropics have more melanin, and people that live in the north or the south have more white, I guess, to them. Or
Less melanin.
Yeah. Couldn't think of the word. So yeah, that would be a paralog
Okay.
in these examples.
Okay.
More white. It's not the politically appropriate term, but this is a Baltics class.
That's okay. Hold on. This last slide. What is conserved? Like an an amino acid that would be considered conserved.
What do you mean by conserved?
Hold on. On here I just have polar and non-polar, but that's where I was getting confused during this. Do you just want to look at it?
Yeah,
We're talking about like alignments and like are conserved. But then down here I have it like a little bit like a question. It says no structure in groups, know what sequence alignment means. Oh, this is just for the exam, I think.
Oh, yeah. So when you're doing the codons, I know exactly what he's talking about.
Okay.
You might have like G, T, C. Look at the codon chart. There's multiple codons. I'm just gonna put multiple codons that code for the same amino acid.
Okay.
So the difference between valine and alanine, is that the example he put? It'd be a change in one amino acid. Sorry,
Okay.
one nucleotide. Sorry, is the word. Like.
One letter.
Yeah, one letter. Like.
Okay.
Like cytosine, thymine, uracil, and adenine.
Okay.
So that's what he's talking about. So that's why it's important that I think it's a histidine to valine change. That's what causes sickle cell anemia. and hemoglobin. So that's what's important of that is.
Okay.
That's what he means by conserved.
Let me go back over here.
All good.
So loud.
Yeah.
Okay.
Uh-huh.
Just talking about like folding.
Yes.
When I was like looking it up earlier because we had talked about it, but I kind of forgot what it was talking about. It's saying that it's forming into like from just a chain to a 3D shape.
Yes.
But wouldn't that be tertiary, but like secondary folds? But it's just locally.
Yeah, that's a great example. So yeah, that's
So, like,
something
how
we
does
need to
that
talk
work?
about. Let's talk about that. Remember, that's definitely important. It's very important concept. Okay, yeah, so primary is just a straight chain
Yeah.
of the amino acid. Secondary, all you need to know is alpha helices and beta sheets.
Okay.
Tertiary is the interaction between multiple of these, so I would say it's more 3D shape,
Okay.
but this is the important part. Single,
Protein chain.
yeah,
Meaning what? Can you give an example?
like it has to be on the same chain.
Okay.
Like if this is one protein chain
Yeah.
and this is another protein chain, the 3D shape of this, this looks like a beta sheet, for example.
Yeah.
That is the tertiary structure.
Okay.
But if I was to add another one like this, or more, it'd be more like this on a beta sheet,
Yeah.
then that's quaternary structure.
Okay.
But beta sheets, so the structure of the beta sheet that looks like this is still secondary. Then we go into tertiary, and then quaternary is multiple. So quaternary has to be more than one protein chain. And we can, let me just draw an example out and show. Okay. So, let's read the pictures. Cool. Pause it there, take a picture. So let's just say I'm drawing this amino acid. And then this is one single amino acid. Just like it's just a line, that's, you don't need to worry about anything else.
Okay.
So there's going to be disulfide bridges here, and then like a disulfide bridge here.
Okay.
So these are all, this is secondary structure. But overall, this, whatever thing this is 3D structure. This is the beta sheet. Anytime you see an arrow, that's a beta sheet.
Okay.
That's what it's referring to. So in this area, we have this activity going on.
This would be considered a parallel beta sheet.
Well, you need another one and opposite running at the same time.
Okay.
So this is just a single beta strand. If it's like this, it's anti-parallel. If the arrow was flipped, it'd be parallel.
So if it's going the same way as the other arrow, it's parallel, and if it's flipped,
It's anti-parallel.
Okay.
Yeah. So like this part right here would be the alpha helix. See, this is the beta sheet. This is another alpha helix. But overall, this is the tertiary structure. Now, if there were two or more of these interacting with each other, that's quaternary.
Okay.
So the best example that I can think of a quaternary structure is heme. Heme looks something like this, which is the component of hemoglobin. I'm just gonna draw octagons or whatever shape comes out. So yeah. not accurate, but there's iron at the center of all these.
Okay.
So these are 4 different subunits. So a whole tertiary structure could also refer to a subunit put together to form this quaternary structure. So all this massive thing is hemoglobin. But in humans, we have to have 4 of these per hemoglobin. Each of these individual ones is called heme.
Okay.
Makes sense?
That makes more sense than
Yeah.
the other.
Well, yeah, that was a tertiary structure.
Yeah.
But then inside that, we have the secondary structure.
Okay.
And inside that, if you looked at it, it'd be like serine, threonine, cysteine, etc., etc.
Okay. I saw that parallel sheets are weaker than anti-parallel, but that's confusing to me.
What was
Because
his
it says that the
reasoning?
hydrogen bonds between strands are angled, and then in anti-parallel they're straight, which makes it more stable.
Yeah,
But when I was looking at the picture, it looked like the
kind of like this.
yeah. Well, yeah.
So this is a parallel, and this is an anti-parallel. I'm just gonna put dot, you get the point. So what's confusing about it?
Why is anti-parallel stronger? Like, why does it? Why
It
does,
takes more energy to break this than these.
Okay.
That's why the angle matters. So this is at a 180-ish, or straight.
Yeah.
Or not at an angle. Not an angle. Meaning greater energy to break. Which makes it stronger. These are bigger sheets. Okay,
That makes sense. I just needed to see it drawn out, I think.
for sure. Okay, anything else?
Talking about how chains can rotate in polypeptides, but
That just comes down to chirality, right?
yeah.
If it's chiral, the whole definition of chirality means it can rotate about the bond.
Yeah.
So that's why there's L amino acids and D amino acids. Like,
Levo and dextro.
yeah, sure, that's how you study it.
That's what was on our study guide.
Okay. I'm pretty sure the L. Yeah, so we have. And the R group, and then the D amino acids. It's going. The H is coming out, and the R group is going into the board.
Okay.
So this is L and D. That's why L is more common, so that it can be facing outwards, making the interactions happen, whereas D is not as common. The only place I've seen it is in
Those bacteria.
transglycosylation in bacteria and E. coli, yeah, which is adding on more sugars.
Okay.
Yeah.
That's it, I think.
Okay. Awesome. What time are we at?
7:19.
Okay, we can finish up early if you don't have anything.
Yeah.
Cool. Gives me time to upload this.
Thank you.